Good morning, everyone. Like that. A warm welcome to all of you joining us today for this exciting webinar on wildlife health in Asia. My name is Daniel, and I am representing Life Trust India, which is based in Coimbatore. It's truly an honor to have such a passionate and dedicated group of wildlife veterinarians, researchers, and conservationists with us. We have more than 200 registered participants for this webinar from around the world, and we are using the different platforms. Wildlife health is a crucial pillar for conservation, especially in a region as biodiverse and dynamic as Asia. The health of wildlife is deeply intertwined with the health of ecosystems and ultimately our planet. Today, we have an incredible opportunity to learn, share, and collaborate on some of the most pressing issues and innovative solutions in this field. This webinar aims to shed light on current challenges, advancements, and opportunities in wildlife health across Asia. We will be hearing from 3 leading experts in the field who will share their insights, experiences, and research. Whether you are working on the ground, in the lab, or in policy, we hope this discussion will inspire and equip you with new ideas and tools to advance your work. Please feel free to just share your thoughts and questions in the chat box throughout the session. We will be happy to just address as many as we can during the question and answer sessions. Your participation is what makes these discussions relevant. I would like to let you know that we will be recording today's session. The recording will be made available for listening after the webinar. And the link will be shared with all the registered participants. So if you miss any part of this discussion or would like to just revise it, you will have opportunity to do it. Once again, I just thank you for joining us. Now, on behalf of Asian Elephant Support, I request Dr. Heidi Riedel to give an introduction about the Asian Elephant Support and also to just introduce the speaker. Thank you. Over to Heidi now.
Thank you, Daniel. Thank you. I appreciate it. And hello to everyone, whether it's good morning or good evening, depending on what part of the world you're joining us from. I would like to welcome everybody on behalf of Asian Elephant Support, who is co-sponsoring these webinars along with Life Trust India. This is a topic, veterinary expertise has been a topic that Asian Elephant Support has been interested in for quite some time. And we have organized a number of workshops and also webinars, not just on elephants, but on other Asian species. So today you're going to hear some presentations that are a little bit broader in nature. And so without wasting any more time, I'll introduce our first speaker. Our first speaker is Dr. Meenakshi Nagendran, who is a wildlife conservation biologist, an academic, and a veterinarian. Dr. Nagendran is currently an adjunct faculty at Cuesta College in California. She retired from the U.S. Fish and Wildlife Service in 2023. At the Fish and Wildlife Service, she worked as a wildlife biologist from March 2005 in the Division of International Conservation, as a program officer for the Asian Elephant Conservation Fund, followed by the Rhinoceros and Tiger Fund, and the Great Ape Conservation Fund for Asia. Prior to joining Fish and Wildlife Service, Dr. Nagendran was in academia in California and served as Director of Bird Conservation in the California Audubon. For many years, she collaborated with international partners on field research and conservation activities in Siberia, Kazakhstan, Japan, the Russian Far East, India, US, and Bulgaria. Many of the projects included satellite tracking several species of endangered cranes, storks, and waterfowl, documenting their migration routes for the first time. She received her bachelor's in science from India, her master's from South Dakota State University in wildlife science, and her PhD from North Dakota State University in zoology, both behavior and conservation biology. And she carried out postdoctoral research at the University of Tokyo. Dr. Nagendran received her DVM from UC Davis in California. Wildlife conservation and ecosystem health continue to be central to Dr. Nagendran's interests, and her presentation today is on the topic of disease investigations in a wildlife conservation context. So Dr. Nagendran, over to you.
Thank you, Heidi. Will Daniel be projecting the slides for me, or should I do that myself?
Maybe you can just do it from your side.
Okay, let me see.
Yeah.
Let me see. I don't want to mess it up. Okay. Does that look right?
Yes, we can see it.
Yes.
Okay.
Start the
Cool.
slideshow.
All right. Yeah. All righty. Thank you. Hello, everyone. So today's presentation from my side is going to be more from a generalist approach. And give me a second, please. I need to make this screen smaller for me. Okay, yeah, that works. Shoot. And what I'm going to be talking about is many things related to investigating wildlife diseases or wildlife events that occur in the field where your approach would have to be more like a forensic scientist almost. So typically with domestic animals, you would follow a case, you take a methodical approach. You would do your subjective, your objective, your assessment, and your plan. And that would also probably be there to some extent, but you need to do more when it comes to wildlife disease investigations, simply because these wild animals are in the public domain. They are most likely managed by a government authority. It could be endangered species. It could be a case where there may be some illegal activity that has resulted in their death. So the records you maintain, the detailed notes you take, the chain of custody that you must keep, these are all very, very important. So you can't just be a veterinarian, but you also need to be looking at the possibility of this going to court. There is the aspect of helping animals that are in difficulty trying to survive, but then there's the aspect of looking at many dead animals that potentially could end up requiring the courts to decide what's going on. If people are present in the area, you need to ask them a set of questions. You need to ask them many questions. And that would give you some idea of what might be going on. If nobody is present, then it becomes even more challenging. And so the way to approach some of this, and perhaps many of you have already done this, and this is not anything new for you, so I apologize if this is redundant, but oftentimes when you get in the field there, you know, if nobody's there, then you need to be thinking about what is it that I need to take samples of. It becomes very important that as a veterinarian who works with wildlife, that you also establish collaborative relationships with wildlife biologists, wildlife ecologists, environmental scientists, policymakers, et cetera. It's really, really important. This is not just a fancy or, you know, this is not just a research project, but this is an investment for identifying potentially what could be causing a very major problem. The questions to ask there are lots of questions to ask. What are the species that are dying? How many have died? When did they first appear? When is there any kind of age, sex predilection? What is the signalment? What are the symptoms of the dying animals? If they're still dying, is it possible that you can tell? Is there any kind of pathognomonic sign that these animals are indicating they're dying from? Are they dying slowly? Are they dying fast? And another very important thing potentially is, are the carcasses scattered or are they all concentrated? So, and as I continue along, I'll tell you some of the, it'll be more of a story form. So this is not my work. I'm presenting from other people's work. So it's just very interesting how things have been identified. And another thing is, if you see something that looks very novel, then you want to see, has this occurred elsewhere? That is really helpful to look at the literature and see, or even just from hearsay, has someone else seen this happen somewhere nearby, far away in another country, in similar biogeographic region? Then, did the animals, was this a toxin possibly? Were they struggling? Was it a neurotoxic situation? What might have been some of the climatic conditions before this event? Was there a freeze? Was there a hot spell? What are the habitat conditions? And what does it look like in terms of, does the vegetation look like it might be, is there any poisonous plant around there? More questions to ask. What were they eating? Did they eat something somewhere and come and die here? Water source. It might seem like, oh, the water looks clean, but is there some kind of a toxin that perhaps was upstream and it came down, and that's the result that, a whole bunch of fish are dying here, but this water looks clean. So what does that require you to do then? Did somebody spray the area? Was there any kind of land clearing? Now, with respect to poisoning, was it intentional or unintentional? And we have so many stories of intentional poisoning, and then so many incidents of non-target species dying. So that's across the world, you see that. What about domestic animal disease? Okay, so there are lots and lots of things, but also remember, if there are people there, get their opinion too of what they might think is going on. And that'll help you as well. And then you may also be formulating some kind of an opinion. So be sure to register those things. If your animal is small enough, you should take the entire animal, collect the entire carcass. Of course, if it's really large, like an elephant, you're not going to be able to take it. So field necropsy, field sampling is really, really important. I also wanted to tell all of you right now that, again, perhaps you know all of this, and so you don't need any of my slides. But if you do, I am providing it as a PDF, and so you can have that as well. So you don't need to take notes. What are the samples that you need to collect? Well, here is just a general slide of what you might need to do for histopath, for toxic, for micro, for parasitology, hematology, etc. And I did want to indicate that when I say a quarter inch, this is quarter thickness, that's quarter inch thickness. What are some of the standards that we use? And basically, the reason I put this together was, and I all got it from elsewhere, it's not my own, is when I first started collecting samples, I wish I had had access to something like this, because it's really important that when you gather samples, that you can use them when you come back to the lab. Otherwise, it's pretty useless. And it's also really important to know how you need to preserve them and how you need to handle them. And you need to worry about not rupturing your red blood cells when you're gathering for serology. So these are things. I wasn't a veterinarian for some of the work that I did at that time, and I was just collecting samples. But as I became a vet, I realized the importance of some of these things. So if you're working with a wildlife biologist, then your skill sets as veterinarians become really important for these aspects as well. Now let's go through some different cases or different scenarios here. For toxins or toxicosis, organophosphates and carbamates are found in pesticides which are acutely toxic, and they're used quite extensively. Some of those are banned here, but they're not banned around the world. And they're poisonous to all animals. And you find that when they're used very specifically for one species, it may not be the case that that's all that is killed. You can have a situation where there are multiple non-target species that are affected by pesticides. So we have poisoning of cattle to, say, kill feral dogs, which doesn't stop there, and then something comes and eats those feral dogs and they die. We've had this happen. I know of cases where vultures are eating poisoned dogs, and the intention was not to kill vultures, but they're eating those poisoned dogs and dying. We've had cases of vultures being intentionally poisoned because poachers did not want the vultures to show the forest guards where their poached animal was. So they would poison after they had poached the elephants. This was this happens, I think it still happens in Africa, elephants or rhinos. They lace it with poison, possibly organophosphates, possibly something else, and you have vultures dying. And so, you have a bunch of dead vultures, but they're discovered much later than you would be able to find a poacher. So these are important situations. You need to know what is going on. It's also very important to keep complete field records, because these records will eventually likely become part of a court case, and accuracy is really important. Chain of custody is really important. Sampling should be done properly and labeled properly. Sometimes you get samples that nobody knows where it was collected, what it is, and it just gets thrown out, and it's a wasted effort. DDT, organochlorine, the kinds of tissues you would need to take. We've had, basically it's a fat-soluble, lipid-soluble toxin, pesticide. And there are lots of ramifications. It results in eggshell thinning in raptors, and for decades this was going on without people knowing what was happening. And then eventually it was discovered that this was because of the biomagnification of eagles and falcons consuming other species. And so basically got magnified along the food chain and resulted in thinning up eggshells, and eventually it was banned in this country. But it's not, I don't believe it's banned in other countries, so this problem perhaps still persists in other countries. Oil spills. This one I have particular experience with, and it was pretty devastating. I was deployed on the oil spill work that happened in 2010 in the US, the largest spill that we experienced, the BP oil spill, and it was pretty devastating. So you've got oiled birds that can't fly, that are constantly trying to get rid of this thing that's coating their feathers, and it's toxic. So we were gathering a lot of the birds, pelicans, all kinds of waterfowl, shorebirds, and of course there were many dead ones too. But then you bring them to a rehabbing facility, or other veterinarians who would be doing with it. They had the task of cleaning the birds with just what they typically use in this country is Dawn dish soap, and making sure that they nourish the birds, they provide them with supportive care. So we were doing a lot of that, and there was just enormous amounts of oil and on the Gulf there near Louisiana. But what was very interesting to me, I spent 5 weeks every day from dawn till dusk working on this project with literally dozens of other biologists and from across the country. And the reporting, the gathering of data, because I was a government employee at the time, and was, it was really important. The government was going to bring this case against BP. It was very, very important that we make sure that when we gather the data, that whether it's a dead bird or dead eggs or whatever, that we meticulously document where we got it from, the time of day, and make sure that the labeling was correct, and make sure that it was put in its own individual packet, and make sure that the chain of custody was maintained, because this was going to be part of the case, the court case that would then eventually, if it was true that all these birds and these eggs, etc., had died from this particular oil that was leaking from the well, they would be fined. And there was a huge criminal case, and it was a huge payout. But we were part of the team that provided the evidence. And for us, it was really crucially important to maintain everything very meticulously. I recall one evening while we were checking nesting islands, and for 2 or 3 days at a time. And one evening when I reported up to what they call the incident command, and it was everybody was talking about what it is that they had done. Not everybody was involved in biology. They were doing different things. There were oil people there. And so when it was my turn, and I had to report what we found dead, all the different things, and I said, I think about 600 eggs, then there was dead silence in the room. And everybody turned around to look at me like, who is this creature that came up with this number? What are they talking about? What the heck is that? And it was very surprising for me, because the days before then, nobody cared what I said, because it was like one bird here, 2 eggs there, and all of a sudden it was like 600 eggs. And they were all part of the case. It went up to our forensic lab, the National Wildlife Forensic Lab, and they had to then get the signatures for the oil to make sure that the oil that had been responsible for that many eggs to die or not hatch, wild bird eggs, were indeed from this spill. So you can see the importance of making sure that everything is gathered in a way and maintained in a way that data are not compromised. Of course, in cases where you have animals that keep going back to the area, one of the simplest ways is to just haze them, to get them not go there. It's really difficult to contain spills in large bodies of water, especially the ocean. So talking about aflatoxins, mycotoxins, aspergillus, this is a big problem. It happens often in many places in the world where you have grains, peanuts, birds that feed there, large numbers of birds eat it and they do die. And so this is something that, if you let grains just stay on the ground and it's moist and warm, and so these are cyclical. This happens, but it's important to recognize what it is. Algal blooms. This is a very interesting situation. So these are also, they go through periodic cycles, and warm weather can create the situation where the bacteria produce these toxin, the cyanobacter, and it can be deadly for all sorts of species, and mammals in particular. We've had in the US, dogs might go on, people's pets may swim and eat stuff or even drink the water in some of these areas and actually get very sick and die. We've had birds actually, obviously, die as well. And it's very fascinating. This was several years ago. I think it was in maybe 2012 or 2013, when more than 300 elephants in Botswana were found dead all of a sudden in an area which they were all, I think it was in one little ponded area or wetland. And the debate was going on, what could it be? What could it be? What could it be? And so a situation like this, it's really important when you discover the animal that you do a lot of environmental sampling as well. Eventually, yes, they said it was cyanobacter ingestion and toxicity that killed over 300 elephants. I believe something similar happened in Nepal as well, where they had several rhinos, not this many, but several rhinos died from cyanobacter toxicity as well, I believe. But again, you might be just a specialist you might be a veterinarian, but without any experience in sampling the environment itself. But you don't have to be an expert to work with others. You can establish collaborations with, especially if you're associated with the university, you can establish collaborations with a biologist or a environmental scientist, or you need to make sure that you get all the relevant samples so you can come up with a proper analysis of what's going on. Again, in the US, there are times when they will post signs to indicate to the public that the waters are dangerous. They also tell you, for example, if you go shellfish fishing, like oysters and things like that, that certain times of year it's very dangerous. Don't harvest it, don't eat it, because you will die. And so that's a problem. This is not just restricted to the US, even though I'm telling you of cases here, this is worldwide. There's another toxicity that's actually very common here, domoic acid toxicity, which leaves animals disoriented. You can have, a sea lion show up in your backyard several miles away from the ocean because it's consumed these toxic shellfish, and they don't know where they are. There's seizures. This happens with birds too. So these are all cyclical again, and potentially dangerous, and at least for people, you need to be aware of it. And if you start to see animals thrashing around, be aware of something like this might be going on. And here, parasitic pathogens, there's Toxoplasma gondii, which is the definitive host is cats. And this is also worldwide. And we've seen Toxoplasma impact otters, sea otters in the US, actually result in their death. And so marine mammals can be an indication, a good sentinel of Toxoplasma gondii contamination. And you can also find this elsewhere in the world. Toxo is problematic for pregnant women. They could abort. So, and it's found in domestic cats. Here it's primarily also feral cats that are roaming around. So they do the toxo. Oocytes are survive a long time, and they don't seem to disintegrate. So we got multiple viral diseases, and I don't think I want to get into all of that, but there are just tons of them, clearly. And they, some are more dangerous than others. And then there's our classic bird flu now, H5N1 in particular, which is fatal to most species, birds and mammals. And here we're starting to see that it's, you've got large numbers of marine mammals dying from bird flu. You've got seabirds dying from bird flu. You've got birds of prey dying from bird flu. You've got people getting sick from bird flu. And it's also reached Antarctica, so it's everywhere. And unfortunately, it's a huge problem. And so the cycle is being well maintained. And I do have to say that, the highly congested environment in which in this country in particular, we practice our agriculture, where cattle and chickens are all kept in large numbers and enclosed areas. This is a great way for it to be transmitted between animals and for them to get sick. And now we've got people who handle them get sick. And so unfortunately, they don't want to use vaccines for this. So it's becoming it's a big problem. Anthrax, and I don't think anybody in Asia is unfamiliar with anthrax. So yeah, it's a problem. There's a way to prevent it. Unfortunately, you can't do much with wildlife. You can't vaccinate all of them, but certainly I suppose you could selectively vaccinate a few animals if they're critically endangered. But sanitation, especially if you've got animals in conditions or in captivity, then you could certainly vaccinate for anthrax. There's an interesting case here, and I wanted to share this. This was fascinating. I was at this lab, and I was asked if I could tell what these birds died from, what these geese died from. And it was really impossible for me to look at it and say, no idea. I couldn't tell. And I just thought, well, was it a toxic situation? Was it toxicology? Or was it, what was it? And interestingly enough, these geese had been struck by lightning as they were landing on this lake. And there was no way to tell other than using this alternate light source. And it was a UV light and it was fascinating. You could tell by shining it on the wing feathers that it was not obvious. So it took them a long time to figure out. So there's another thing, you find a whole bunch of dead animals, but you can't really tell what it was because the surrounding area looks fine, but perhaps they came in when there was a lightning storm and that's how they died. And So be prepared for something like that as well. And I will end this with the case of zoonotic diseases where species jump, where disease pathogens jump from species to species. And I'm just been following or watching this workshop. This is just wonderful. And if you have the time, it's multiple hours of zoonotic disease, patient zero, preventing patient zero. And if you have the time, I would encourage you to watch that, and hopefully you have access to it. It's through the National Academy of Sciences. And they talk about how complex it is. It's easy to say that, we need to go in there and get rid of this wildlife and get rid of that wildlife, or this is the problem. But when you listen to it, you'll realize that it's not so easily done, not so easily defined, not so easily diagnosed. And the complexity of disease dynamics and disease ecology is fascinating for sure. So on that note, I will end my presentation and take any questions if you have them now, or
Yes, Mini, we have a few questions. I think
sure.
one question. Thank you so much for the presentation on disease investigation in wildlife conservation context. There is one question from Dr. Azad. Can we use hematological machine used for domestic animal or pets for wild or zoo animals, or is a specific hematology machine for zoo and wild animals we use? Yeah, that's a question.
I think you can. I think it's just, if you, for example, I believe if you had if you were looking at, say, if you used a machine for chickens, you could certainly use that for other avian species. So there are, I think it's just the settings. Absolutely, you could use some of those machines. They're not very specific to a species per se. Not all of them.
Yeah. Okay. So, is there any questions?
Well, thank you.
Yeah. Thank you, Dr. Mini. Now hand over the session to Heidi.
Okay, thank you. And thank you very much, Minnie. We appreciate your presentation.